for joining our webinar series. We are looking forward for today's webinar, Identifying Structural Variants in Mendelian Diseases. I would like to start with a technical introduction because it's an online event. However, we would like to be interactive and we are looking forward for all your questions, comments, criticism, and we are really happy if you address all your concerns and questions in our Q&A session. I will do my best to answer each of your questions, and whatever we don't manage to answer, please contact us directly. We will always support you. So you have, it's a Zoom setting, you have a button Q&A that you can press. Afterwards, there is this appearing, and here you can type your question, and then you can just simply press send, and then the question will be delivered to us, and we will answer this question, as in this example. So, and then some questions we will answer immediately about some clarifications, and some questions Najim will address directly. And in the end, whatever we don't manage, we will always contact you and we will answer your question. And today, I'm very happy to introduce my colleague and today's speaker, Dr. Najim Ameziane. So Dr. Najim is a Senior Director of Bioinformatics in Census. Najim has a PhD in human genetics with years of experience in molecular diagnosis of hereditary diseases and cancer. He pioneered the application of next-generation sequencing in the clinical genetic department of the Free University Medical Center Amsterdam. Najim is passionate about the human genome investigation, which holds the code of life. I personally know Najim, and every time there is an unsolved case and the patient has a genetic disease, Najim will find a reason. And we are looking forward to your webinar, Najim. And I will briefly explain after the webinar, our next webinar, and how to get a certificate. So I welcome Najim for his webinar, and we're all looking forward for it.
Thank you, Bolha, for the kind introduction and the nice words. And I would like to welcome everybody to this lecture. And I hope you enjoyed. And as mentioned earlier, if you have any questions, please send them to us, and then I will try to answer them as best as possible. So let me share the presentation with you. Okay. So in this webinar, I will try to explain to you what structural variants are and how they are involved in a Mendelian disease and how these types of alterations can be identified. Okay, so in this presentation, firstly, I would like to introduce the concept of DNA testing. As you know, whenever we do a DNA test, we try actually to translate information that is biological, so some biological elements that is eventually transformed into some digital information, and finally to information that is readable to humans, which is eventually provided in a report. Furthermore, I will go a little bit into details with regards to the type of variants that are known to cause disease and how these variants are detected using next-generation sequencing. And then I will show some examples, and finally I will leave it open for questions and I will try to answer those. Okay, so when a patient sees the doctor and the doctor suspects a genetic disease, a DNA diagnostics test is then proposed. And usually what happens, a biological material is taken. Often this is done by extracting blood or buccal swabs, which is the easier way of doing so. And when extracting this material, eventually what you have extracted is a bunch of cells. And from those cells, we extract DNA, which is the second phase. And once the DNA is extracted, the data is actually from the genome is read using next generation sequencing. And with this way, we try to convert this biological data into something digital that can be assessed and checked very quickly and fast. And then an intervention with an expert looks into this data by identifying and trying to identify the relevant information that is associated with the condition of the individual. And this is then translated into a report that can be read by the physician or directly by the customer who is interested in this data. So first of all, when it comes to DNA testing, there are some things to be considered. The first of which is that there are different modes of inheritance. When it comes to heritability, information that can be inherited from parent to offspring, it can happen in different ways. So when it comes to disease, the disease can be inherited in an autosomal dominant manner, meaning that the parent is carrier for the disease, and also the symptoms are manifest in the carrier. And this carrier can then give this mutation or variant that is causing the symptoms to the offspring. And therefore, the disease is called an autosomal dominant disease. And then you have an autosomal recessive mode of inheritance. Here, the carriers who are the parents, they do carry a variant that is indeed explaining the symptoms. However, the carrier themselves are not affected. Only when both carriers give the disease-causing alteration or the condition-causing alteration to the offspring, that's when the manifestation of the symptoms happen. And then there is the X-linked dominant. That's when the disease-causing or the condition-causing alteration is present on one of the 6 chromosomes, specifically the X chromosome, which is inherited from the mother. And therefore, since the alteration that is associated with symptoms is on the X chromosome, all the individuals who carry the X chromosome will be affected. And since it's dominant, both males and females will be affected. And then the X-linked recessive disorder, that means that both X chromosomes need to be affected to cause the disease. And since males have only a single X chromosome and they don't have the other wild type X chromosome, all the males will be affected, while the female carriers or the mother who has initially this particular alteration or the variant is not having the symptoms. And then there are also diseases that involve the mitochondrial genome, which I will not go into much detail in this presentation. So besides the mode of inheritance, there are different types of variants that can eventually lead to symptoms or a condition. This can be divided in 2 main sections. The first one being the subtle variants. And these are alterations or changes in the genome, in the DNA, that affect up to 50 nucleotides of the DNA. On the other hand, we have the structural variants, and these are changes or alterations affecting large chunks of the DNA. And these alterations range, I mentioned here that they affect more than 1,000 nucleotides, but they can be also a little bit smaller. But in general, they are much larger than 1,000 nucleotides. They are more in the range of kilobases, so hundreds of thousands of nucleotides, or even millions of nucleotides. So the disease-causing variants, as I mentioned earlier, when it comes to the subtle variants, of which I will show examples at a later stage, this can, on their part, be divided in 2 sections. So these are the single nucleotide variants and the small indels. Single nucleotide variants are actually changes of one nucleotide into another. As most of you, if not all of you, know that when it comes to the DNA, there are 4 letters that are making up our DNA, the A, C, T, and G. So a single change from an A to a C, or whatever change possible, is called a single nucleotide variant. As for the small indels, these are deletions or insertions that involve less than 50 nucleotides. These type of subtle variants are routinely examined and checked in genetic diagnostic setting, and the sensitivity and the specificity of this type of analysis exceed 99%, which means that it's extremely accurate. On the other hand, we have the structural variants. So these are, as mentioned earlier, alterations that involve more than 1,000 nucleotides. And most of the time they involve even larger sizes, larger than thousands of nucleotides. And one has to keep in mind that these types of variants account for about more than 10% of the diagnostic yield when it comes to genetic testing. So this is a significant number of alterations that can explain syndromes. And however, when structural variants are assessed, it is accompanied with a high number of false positives. And that's one of the reasons why it is not applied widespread by many companies. Nevertheless, since it accounts for a significant number of disease-causing variants, it is important to address those in routine diagnostics as well. So the different types, if we dig a little bit deeper into these types of variants, again, the subtle variants are these single nucleotide changes, and these deletions, insertions, this involve less than 50 nucleotides. And when it comes to the structural variants, we can deal with large deletions, so large chunks of DNA that are deleted. They are no longer present in the DNA of a cell, or large insertions, which can also be large duplications, so segments of the DNA that are duplicated into the genome, which can also cause disease. And all other types of structural variants are inversions and translocations. I will attempt to explain those in details at a later stage and show some examples as well. So just to have everybody on board and to give a simplified explanation about types of variants. So consider the sentence here at the top. Everybody can read this. And as a matter of fact, I attempted to use the 3 letters per word, which is usually also what we see in the DNA, because the code that is used by the DNA, by the 4 letters that I mentioned earlier, are usually coded in a way that they actually code for pieces of the proteins based on 3 letters of the nucleotides. So the top sentence reads, "The fat red cat ate the wet rat." When we are dealing with nucleotide changes, one letter, in principle, can be changed and can alter the meaning of a sentence. So when we are altering the C in cat into a K, in principle, we do have a mutation or a variation. However, the meaning of the sentence, although the spelling is incorrect, so we are dealing with a substitution, but the message is still understood. As for the other nucleotide change, as in the sentence that I highlight right here, it's also a substitution. However, since the C is changed to an S, the meaning of the message is no longer clear to whoever reads the message. So this type of mutations are also called a nonsense mutation. And then we can have a deletion that actually deletes only a single letter, which is this T over here, the T of the cat. And since the T of the cat is deleted, all the downstream letters that come after the cat, they will be shifted to the left. And therefore, because this shift has taken place, this mutation is a deletion. It is also called a frame shift. And due to this frame shift, the sentence can no longer be accurately read, and the message becomes a nonsense message. And the same thing is true for an insertion of a single nucleotide, where we have a T inserted after the T of the cat, although the cat still remains the same and is understood by when it's read. However, what comes downstream is no longer readable because all the other words will have an extra letter added and the whole message will be shifted. And this becomes also a nonsense message. On the other hand, when we are dealing with structural variants, a large chunk of the DNA can be affected. For instance, complete words or complete sentences can be deleted, which will also result in a nonsense message. And the same thing is true when there is a duplication that is duplicating either an entire word or a sentence. This causes a nonsense message as well. And the same thing is true when we have an inversion, when we have a piece of the message inverted and inserted back into the same sentence, the message becomes nonsensical. And the other examples involve translocations when we have a piece of one chromosome that is broken attached to yet another chromosome, and this is similar in a similar manner would render the sentence unreadable and nonsensical. So the two types of grouping of categories of alterations are single nucleotide or small indels, which are the subtle variants, and then the lower part, these are the examples that are called the structural variants. So this is just a visual illustration of the types of alterations that can be present in the DNA and that can be associated with disorders and conditions. So we have on the top panel, right, what that we see here is an illustration of the reference sequence. Whenever we have a change from one nucleotide to the other, we can view this by reading this sequence, and then when a difference is identified between the reference and whatever has been sequenced in an individual, this is then called a single nucleotide variant, also known as a point mutation. When a piece of the nucleotide or a piece of the DNA that is smaller than 50 nucleotide is deleted, then it is viewed as such. There will be a gap in the sequence that has been sequenced. And when we are dealing with large parts of the DNA that is affected, then a huge part of the DNA will no longer be sequenced and therefore no longer be existing in the subject when comparing to the reference. And if both alleles, so both pieces of the chromosome from the mother and the father are affected, then we are dealing with a homozygous deletion. And when only a single part or a single allele from one of the parents is affected, then this is called a heterozygous or hemizygous deletion. Similarly, a duplication or a duplication of a segment of the genome is called a gain, when there is more information available or more data available than in the reference. And when we are dealing with translocations, as I mentioned earlier, a piece of a different chromosome is broken and attached or ligated to another piece of chromosome. And with whole genome sequencing, we can indeed also identify DNA that is not coming from the human sample. As you can imagine, when a sample is extracted from an individual, it can also contain cells from microbial species. This is often mainly the case when buccal swabs or saliva is sequenced, as the oral mucosa may also contain bacterial microbes. So the types of mutations that are subtle, like the single nucleotide variants and the indels, they can be easily detected by all types of NGS methods, including whole genome, exome, and panels. And when it comes to the structural variants, and mainly the CNVs, the copy number alterations, which are deletions and duplications, this can be also detected with exome. However, the detection limit is less, and when the deletions are below a certain range, they no longer can be detected with exome. And on the other hand, when we are looking at structural variants that cause translocations, these are the type of alterations that cannot be detected with exome. However, all these types of alterations can be detected with whole genome sequencing. So just briefly, an explanation on how NGS takes place. So when the DNA is isolated from a subject, which eventually would have DNA in a tube that consists of large molecules that can range from 50 to 100 kilobases. So these are 100,000 nucleotides long, up to 100 nucleotides long. This DNA has to be fragmented into smaller pieces, as the technology that allows the sequencing of these fragments requires that the fragments are within that range. And this range of the DNA that I'm talking about is based on the most widely used NGS technology. However, there are other technologies that can also sequence much larger fragments. But I'm focusing on the sequencing technology that is mainly based on Illumina sequencing that only sequences smaller fragments. And once this DNA is shared to a size of around 500 bases, then adapters are added to each side of the DNA, so eventually it can be sequenced. So the assumption when sequencing is taken place with this technology is that the fragments of the reads that eventually get sequenced, they would have a certain size based on how fragmented this DNA is. And I will come to this later. Once the DNA is fragmented and the adapters are added to this fragment, then these fragments are then put on a sequencer. And what takes place first, images are taken from these fragments after introduction of nucleotides, which is done in cycles. And once these images have been taken after so many cycles, which eventually specify the size of the reads that eventually will be obtained, and these DNA fragments are called the raw reads. So these raw reads contain information about which sequencing machine has been utilized and the actual nucleotides of the fragments that have been sequenced. And once this information is obtained, it will be put in a format that is called FASTQ file. And these FASTQ files are then aligned, which can be done by multiple types of tools. And this is usually done on large servers because it consists of very large files, especially when we are dealing with whole genome sequencing. And once the alignment has taken place, and the alignment, what happens at that stage is that these particular fragments, which are typically 100 to 150 nucleotides long, they are mapped back to the reference genome to identify at which location they came from in the first place. And by aligning them to the reference genome, we rebuild the genome of an individual. And once this alignment took place, then the variant detection analysis takes place. And at this level, there are different types of detection methods. So for the single nucleotide variants and the small indels, so the subtle variants, there is specific tools that can execute this analysis. As for the structural variants, there are different types of tools that can execute this analysis. And for this, also a different type of VCF files will be created. Once we have the variants, then we can do the annotation to provide context to the data. So now I need to go fast because I still have not so much time left, I see. So when it comes to structural variants, we have two different methods to identify those. There is either a quantitative, which is based on DNA dosage. So how many copies of a specific segment do we have in the genome? And this requires the interrogation of multiple samples. When you want to execute this for the CNV detection, you would need the analysis of multiple samples at once. As the other method, which is based on a qualitative analysis, and this qualitative analysis considers the size of the fragments that I mentioned earlier, where it is expected that the size of the fragments is around 500 bases. And this exploits the information from the different reads that are sequenced. And this can be executed on a single sample. So you don't require multiple samples to be present to execute this analysis. There are different tools that can identify structural variants. Some of them can only identify structural variants based on the read depth. So how deep was the sequencing that took place for one sample as compared to reference samples. And then there's other tools that do the structural variant detection based on the read pair information or based on the same read, from the same read, whether, for instance, a split took place during the alignment, yes or no. So there are many reads, many tools that can do this analysis. So the dosage-based CNV detection, it can be explained as such. When we have a normal situation, we expect a certain depth of a particular region to be covered. If there is a deletion that took place, meaning that we have lost DNA at that particular region that is deleted, then the expectation is that the depth of coverage will be decreased. And this is then identified by analyzing the sample as compared to the reference, which is multiple samples that are expected to be normal for that region. As for the duplication, it's the other way around. The normal depth of coverage is expected to be at a specific height. When there is a duplication, more data will be observed in that particular region, and therefore the coverage depth for that particular region will be higher and a duplication is suspected. So here are some examples of subtle variants. So each time I will show a top a wild type situation, and at the bottom a situation where there is a variance. So in the first example, we see a substitution where there is a nucleotide change from wild type, which is a T to a C, and here we are in an example of a deletion of a single nucleotide, and here this is an example of an insertion of a single nucleotide. So these are the subtle alterations. As when it comes to structural variants, this is an example of a deletion. Each time we have an alteration, we always compare to the reference genome. So if the subject has a deletion that no longer has this segment of the DNA present, once the sequencing of the subject took place, if this piece of information is no longer present, the distance from this end to this end will be brought together. So these fragments, when it's sequenced from the subject and it will be aligned to eventually the reference genome, the distance that is sequenced from the sample that was expected to be around 500 bases will suddenly increase when comparing the forward distance to the reverse, the distance from the forward read to the reverse read. And this way, this reads will be flagged. And how does this look like? So this is an example of a deletion where there's a homozygous deletion. We see, first of all, that data no longer are present for the segment that is deleted. And this is an example of a deletion that involves 104 kilobases. And it affects the two genes are mentioned down here. The names of the genes are at this stage not so relevant. But the homozygous deletion means that there is no data here. And when we are dealing with a heterozygous deletion, then we see still some data present, but it's much less in the region that is deleted. We can see this at two locations. We can see at the coverage plot over here that there is a sudden decrease in the coverage, which goes up again at the other breakpoint. And simultaneously, at the edges where the deletion took place, because the reads, the forward and the reverse read have an aberrant distance between them as compared to the reference, they will also be flagged red in the case of the deletion. I just got notified that I have to speed up because the time is almost over. So for the sake of time, I will just jump over this example of a duplication. I will go into an example that is a complex structural variant, which is in this context most interesting. So this was a case that we have analyzed where we have identified actually what seemed to be initially two separate events. So we observed what looked like a deletion on chromosome 14, and we saw in the same individual something that looked like a duplication of 2.5 million bases on chromosome 15. However, upon closer inspection of the breakpoints of the deletion and the breakpoints of the duplication, we observed that many reads actually, of which the forward were mapping to chromosome 14, their mates, so the mates, meaning the reads from the same molecule, as you remember, we sequence from the forward and the reverse for the same molecule. And this way, you can actually assign colors to those reads, and you will identify their mates once we have given them a specific color. So by doing so, we were able to identify that many of the reads that are at the breakpoints on chromosome 14, their mates were actually aligning to chromosome 15. And this led us to actually confirm, or to have this provided evidence that we are dealing with a complex rearrangement that involved translocation. And what we think happened in this particular example is that a duplication took place on chromosome 15, and simultaneously, or at the same time, there was a deletion that took place on chromosome 14, and the part that is duplicated in chromosome 15 has been translocated and connected to chromosome 14. And you can imagine at the breakpoint where this took place, genes that are involved in that particular region will be abrogated. So by whole genome sequencing and executing this structural variant detection, we were able to provide the accurate annotation of this variant, which is only possible with whole genome sequencing. With exome, you could possibly have identified the deletion or the duplication, but you would not be able to identify this particular translocation. So to conclude this webinar, I want to stress that structural variants are responsible for over 10% of hereditary disorders. So this is a significant number of alterations, and therefore it is warranted to address those types of alterations in genetic testing. Many types of structural variants are exclusively detectable with whole genome sequencing, and this involves very small CNVs that cannot be detected with exome, for instance, and certainly inversions, which are not detectable by Wes, and translocations, of which I have shown this example at the end. And structural variant detection is essential for comprehensive genetic testing, as I mentioned earlier, as this involves a significant number of individuals that otherwise go missing. As a matter of fact, we have several examples here at our company where we sequence individuals that have been tested negative by Wes elsewhere, and we were able to identify the disease-causing alterations by performing whole genome sequencing together with structural variant detection. And with this, I would like to open the floor for questions. Thank you for your attention.
Thank you very much for the great webinar. Before questions, I will briefly explain you the next steps. Okay. So our upcoming webinar is September 28, will be done by Professor Dr. Arndt Rolfs, and it's 7-part. And we always talk about unexpected diagnosis based on whole genome sequencing. And this webinar is created by you. We constantly get requests from you to comment on their solved cases that were negative before, panel, single gene, and Wes analysis, and that we were able, for sure, together with Najim, identify and understand what was the reason of the disease. And these cases Professor Royce present in this series. And an important note that we're happy, and you always apply to get their CME certificate. So it's super easy. And this certificate is only issued to participants who request it using the platform. And please complete your details correctly. So your title, your first name, your last name, so that we issue you a correct certificate. And in case you did some typo, you can always contact us and we will reduce the certificate. And thank you very much for your attention. And with that, I would like to start our questions. So there are lots of questions, and I have a power to choose what to ask. And what we don't manage, for sure, we will do after the webinar. So we have a question from a schoolboy from Lebanon, and he's asking why structural variants are so big, however difficult to identify. Do you believe that there will be a microscope in future which can detect them?
That's a very interesting question. As a matter of fact, there are some equipments that actually behave like microscopes and that can detect a specific type of structural variants. As a matter of fact, some of them are detectable by microscopes, because what we can also do when it comes to specific structural variants like translocations, as I mentioned before, sometimes what happens, these chromosomes are stained by specific dyes that actually stain specific chromosomes by specific dyes. And when having these dyes added to the chromosomes of the cell, a microscope is used to see actually whether the chromosomes actually light up as expected. So for instance, if let's say chromosome 1 has a green dye and chromosome 2 has a blue dye, and all of a sudden you see that at the tip of these chromosomes that the dye goes from green to blue at one chromosome, this would be suggestive of a translocation. So indeed, we do have microscopes to look at some structural variants. However, the majority of structural variants that, for instance, involve copy number alterations, if this copy number alteration is smaller than a specific size, then they are no longer detectable by the microscope. But there are some other equipments that simulate what the microscope sees, and we can indeed use this to identify these alterations. As for other alterations, why they are so difficult to detect in the first place, because the technology of NGS sequencing that is currently used, they are focusing on, they have to firstly fragment the DNA to very tiny fragments. And because the fragments that we start off with in the beginning are so small, it makes it difficult to identify within a single fragment this large alterations. And therefore, we have to utilize the information from these reads that I explained earlier, or executing this quantitative analysis where we look at the coverage. So it sounds counterintuitive. We have large fragments, but nevertheless, they are more difficult to identify than smaller fragments. But this is how biology works.
Thank you very much. What do you mean with small copy number variants? What is the size limit in VGS to be detected?
So the size for VGS, there is actually no limit when it comes to identifying CNVs as to the size, because when you execute whole genome sequencing, you cover the entire genome. And the problems that, for instance, other sequencing technologies like exome or panel sequencing have, that an enrichment step takes place. So you need to enrich for the regions that you are interested in. For exome, for instance, or panel sequencing, the enrichment method usually focuses on enriching the exome, the exonic regions, the regions that are coding. And these regions are usually interspersed, distant from each other. So one exon can be thousands up to millions of nucleotides distant from each other. So if you have, for instance, a deletion that affects 2 exons, although the deletion at the genomic level may be more than 1 million in size, but since with exome, for instance, only the exonic regions are sequenced, the data that is obtained is only focused on those 2 exonic regions. And since the CNV detection on exome data, on enrichment sequencing data, only can try to identify alterations for the region that has been sequenced, and it relies on statistical analysis that involves analyzing the sample in the context of a set of samples. This size, although it's more than 100 million nucleotides in size, when you look at the data that is covering this region, it's still actually very minute. And therefore, deletions that cannot be detected with exome, for instance, but can be detected with genome, are deletions that involve less than 3 exons. Just to give you a rough estimate. And this is usually what actually the majority of diagnostics labs also provide as limitations for exome sequencing. If the deletion affects less than 3 exons, then they may not be detected.
Thank you very much, Najim. We have 2 questions from evolution section. And the first one is, can structural variant lead to advantageous phenotype, let's say like resistant to infections, and not to disease?
So when it comes to microbiomes, microbial species, yes, I can imagine that it can. But as far as when it comes to human genomes and evolution, as far as I know, I don't have any evidence or knowledge about structural variants that indeed can provide any advantages evolutionary. You may maybe, for instance, some mutations that because what you have to keep in mind when we are dealing with a structural variants or mutation, for that matter, in general. Mutations usually affect the coding region that is eventually coding for how to produce a protein. If there is a mutation, usually it will cause the abrogation of this particular protein that has a very specific function. And if, for instance, a large alterations mainly, they are not expected to be evolutionarily advantageous because they are either will abrogate the function completely if you have a large duplication or a deletion or even an inversion or translocation. They are not expected to have a function available. However, in cancers, it can happen that sometimes you have translocations that result in a fusion and where actually 2 pieces of proteins, 2 pieces of DNA are actually brought together, and this will then generate a protein that will have an advantageous effect at the cellular level. But evolutionary, I don't see how this can have an advantage for the organism as a whole, although it may have some kind of advantage for the cell to grow faster, for instance.
Thank you very much. And the second evolutionary question, which for sure will probably require a super long answer, but I would still like to mention it. So abnormalities, if I can divide them into missense, variants, and structural variants, why evolutionary both ways exist?
So I don't think whether the evolution is the explanation for their existence. Actually, the existence of such alterations can be explained by the way the DNA replicates and how this replication process sometimes causes errors, and how the machinery that exists to actually repair those errors evolved during the time. So you have different types of mutations, and you have different types of DNA repair mechanisms that can repair those different types of mutations. So for point mutations, for instance, there are specific methods in a cell, this specific mechanisms that can repair this type of mutations. And for the structural variants, there are yet different types of mechanisms that can repair this DNA. So evolutionary, I don't think that's the source where you have to look for to explain why some mutations happen. But you have actually to look into the mechanisms that are involved in the repair of this DNA to identify answers for why this type of mutations happen.
Thank you. There are lots of questions, but I will ask the last one. Is whole genome sequencing able to determine breakpoints for balanced rearrangements, such as translocations and inversions? And the second one, please tell me how to identify inversions.
So regarding the first part of the question, as a matter of fact, this was the last example of the presentation that I showed. So we had, as a matter of fact, a balanced, in this particular case, it was an unbalanced classification, but similarly, because eventually when you look at the breakpoints, you can indeed identify exactly the reads that are involved in this rearrangement. And you can identify at the nucleotide resolution which nucleotide is involved in this alteration. So yes, we can identify the breakpoints and we can identify balanced translocations. As for the inversions, that was an example that I had actually in the presentation, which I was not able to go into detail, but indeed we can also identify inversions. As a matter of fact, we have examples of inversions that we were able to detect and explain the symptoms. So similarly, when we have a subject that has an inversion in the DNA, the DNA of this individual will be again fragmented and sequenced. And because once we have sequenced the location of the breakpoints of this inversion, and then this data will be aligned to the reference genome, then because the reads will no longer align as expected due to the fact that the piece of the DNA at the breakpoints will align differently, these reads that will align differently will be also flagged in a specific manner. And this flagging will be utilized to identify the breakpoints and the region that is exactly involved in this inversion.
Thank you. It's just the last comment or question that you just decide, how do you comment on this? It's again about evolution. What if we detect multiple types of mutation in one organism, and this will pass to another generation? Can it lead to a new species to be evolved?
Well, it may, but we won't be able to see it. We won't be able to experience this new species, because usually the majority, and I think this is important to stress, the majority, over 99.9% of the alterations in the genome are deleterious. So the chances of observing an alteration that will have an advantage evolutionary is extremely low. And if they exist, even if they happen, then the consequence will be so minute, it won't be even that easy to recognize or to observe. It may take even much larger amount of time to be able to experience the final effect of this evolution. So we are talking about hundreds of thousands of years, if not millions.
Thank you so much. So with that, I would like to conclude the webinar. We try to balance our information together with your questions, but it's very amazing for your interaction with us, and we're really looking forward for more and more questions. And everything what I didn't address today, I will do it. Otherwise, please contact us. And thank you very much. So we are closing the webinar. Thank you.
Bye.